Thank you. I'll share my screen.
Yeah.
Okay. I'll start my presentation. First of all, thank you for the invitation, and I'd like to say my gratitude to KBI and BioSM for this kind of a very useful webinar series. Today I have prepared plant and animal genomics as a brief introduction to the lab process and some of the bioinformatics processes involved. The first topic would be de novo assembly. I'm not sure how many people are interested in de novo assembly, but that is a very important part of the plant and genomics applications that I've experienced so far during my tenure in Microgen. So I summarize the current strategy, so to speak, about the de novo sequencing assembly of the plant genome. And the second of all, making the reference genome is not the ultimate purpose of the genome sequencing. It is what kind of information or what kind of development they can do on the reference genome. So I would like to add the population genomics applications after the de novo assembly. And if I have some time left, I will briefly introduce the other part of the Microgen side. Okay, de novo assembly. Assembling a genome is exactly like solving jigsaw puzzles. But there are many biological challenges that makes the assembly so much difficult. You cannot really, fit the pieces together because, if you need to fit the pieces together, it should have an overlap. And sometimes you don't really have the pieces. I mean, there are missing pieces or the bad sequencing quality. So there are lots of sequencing bias in the genome assembly process. And also some pieces will be very dirty because there will be, inevitable sequencing errors. So even if you are starting with very good data, it is very difficult to get the fully these are the biological challenges for genome assembly. Theoretically, genome assembly is very straightforward, but these are the things that makes the genome assembly so much difficult. The first thing is the lack of reference genome. I'm not talking about the full and complete genome. It could be a very low depth or even transcriptome. That is much, much better than having nothing. So if you don't have anything, very, very new genome, you got to know that it'll take a lot of time and efforts. Then the genomes with which you have some reference to. And the other difficulty is, like a human genome, most genomes are much larger than human genome, especially for the plants. And there are many different kinds of repeated sequence, especially plants and animals, which makes it very hard to, to uniquely map or uniquely align two pieces together. And also, plant has a many multiple ploidy. That means that makes a complete genome assembly so much more difficult than just the diploid genome. Okay. Well, this is straightforward. If you have some kind of a reference, it will be easy to assemble it. But it is really like the solving the jigsaw puzzle without any final picture would look like. So you can guess it is almost impossible to start. And but well, frankly speaking, all the genome sequencing started from this stage. So it will be better if you have a reference, but you can start by piecing only two pieces together. That is the very good start. Plant genome, it has usually have like a 20 to 100 gigabytes range. Human genome is 3 gigabytes. And the left side of the genome, which is estimated to almost 500 gigabytes. It is the amoeba species. On the other hand, a bacterial genome with the size of 1 megabase, you can do it. You can do the whole genome, complete genome sequence in a single circular form in a week. So that is the size really does matter in the genome assembly. Okay. Repeat is like this. You are looking at the picture. If you see the picture, you're solving when you're solving the puzzle, you can easily solve the puzzle with the different colors and different text, and so you ended up with the puzzles that cannot be distinguished from each other. So these are actually, in a biological sense, many different kind of repeats. It could be transposons, it could be satellites, or it could be gene duplications, which makes it the uniquely mapped assembly much more difficult. Yeah, when you are having a very, very long range repeats, it is almost impossible to find the strain graph. That means you are looking at the very long range patch of a DNA, like a string, and it is impossible to find the right the string. So you end up with the very twisted string, which you cannot really solve. And multiploidy is a totally different story. Bacteria only have a haploid. That's why it makes it so easier to assemble it. Plus the size of the genome is small. Most of the genomes are diploid, especially for the human. But the sequencing genome is one thing. Getting the phase information between the two diploid is a totally different story. It takes a whole lot of different efforts. I have one experience of phasing the genome, human genome, and that was very, very difficult. And it took us a lot of different approaches in terms of experiment and the analysis. And I don't have the first-hand experience of the triploid or tetraploidy, but I can only guess how difficult it would be. So that's why, this is the wheat plant, which is a hexaploid. So I admire the authors of this the consortium very much, because I can only imagine how difficult it is to get a phased genome in the hexaploidy organism. Well, getting back to the basic, de novo analysis workflow is very straightforward. You get a raw data, And you start by the genome survey using the k-mer analysis, and you do the assembly with many different kind of assembly algorithm or systems. And after that, you get the draft of assembly, and you start from there and go directly into the gene prediction and annotation. Genome survey is important in a way that you can get the estimate of the genome size. It could be like a gigabase level, or tens of gigabase level, or even 10 megabase size. With that, you can decide how far you should go, or how deep you should sequence the genome. And the second one is you better have a good idea of how many chromosomes this new species have. And with that information, the estimate genome size will be used as a reference for the assembled genome size. In practical terms, we are using the flow cytometry and PCR as the experimental tools. Then we use the databases, different databases, and do the k-mer analysis of genome shotgun data in silico. That's when we come up with the result of the genome survey, which are the estimated genome size, which we can use to determine the sequencing amount, the sequence heterozygosity, GC contents, or even if there is any sample contamination. So we use this one to make it more efficient for the experimental design and make the predictions of the final assembly. Genome assembly strategy is very straightforward. There is no single sequencing tools or method or bioinformatics pipeline that can do this alone. You should have all kind of different experimental system. In other words, different sequencing machines and many different algorithms. So this is a very simplified version. On your left, you will see the short-read sequencer, such as Illumina. And on the right side, you are looking at the long-read sequencing, like a PacBio or Oxford Nanopores. Short-read sequencing, you can go very deep. So that's the reason that you will get accuracy. But because the read is very short, there should be a very incomplete picture. With the long-read sequencer result, you will get simply a bigger picture, but it occasionally lacks the accuracy or the biological replication, or it suffers from more sequencing errors than the short-read sequencings. So it's ups and downs. So that's why we are doing both, and take the good part from each different platform. In a practical sense, we basically get the long read up to 20 kilobase using the PacBio or Oxford Nanopore or other long-range sequencing platforms, and we read up to 150 base pairs of the short read from Illumina. We use them mostly for the error correction for the higher accuracy. So with the long contig and higher accuracy, we are good to go for the complete genome assembly. And after the genome assembly is done, we usually do the follow-up analysis of the novo assembly. Most of the important things, two things, we find what kind of gene this genome is having, and we can basically annotate the genes from the reference sequence. And we can also do, come up with the genome structure and functional analysis with the bioinformatics pipeline and all the databases we can pull. So for the prokaryote, is very simple. But eukaryote, they are much more complex genome structure. So we use different kind of algorithms using the gene model and the transcriptome and come up with a new gene model. So we usually do this one for the plant and animal, or even the fungi. This is the PacBio. The more recent one is Sequel 2. This can produce a very long read. As you can see, the average long read from the Hi-Fi protocol is more than 15,000. And if you are using the Hi-Fi read with the enhanced accuracy, you can get very, very good quality data. So these are the essential tools for the big genome assembly data. But still, genome size is much, much bigger than the average read length of the PacBio platform. So we need something else that can cover the much longer range. So with this BioNano system, you can really take a look at the very, very long range, ranging from like 1 kilobase to up to the 100 megabase range, which actually people or researchers has been assessing using the karyotyping or FISH. So it's a big whole different level of the assembly. It doesn't really produce the sequencing. It just it just helps us to sort the long range order. So like if you have a repeated extension, inversion, insertion, deletion, with a size of more than 100 kbase, you cannot really solve that. But with this BioNano technology, you can order all these different kind of big, giant structural variations. So this is the final slide that summarized my discussion up to so far. And I won't repeat that one. Okay, second topic, population genomics applications focused on the low coverage whole genome sequencing. These are the pictures of the genomes that we have so far participated to assemble. Well, you can see the apple, or maize, or cabbage, things like that. But now what? You really got the reference genome. Now what? But if you really think about it, the reference genome is not the ultimate purpose or the goal. The purpose of the genome studies is the potential for the application is enormous. So these are the things that our the collaborators or our customers has been targeting for. Someone wanted to make a value-added crops, or someone wanted to make a faster horses, or have something with the heavy metal resistance, or hotter pepper, or some crops that resist the extreme weathers. Recently, these are there is actually the world is in the food war again. So these newspaper is actually coming from the Russia, who actually banned the export of their crops. So, and this is the example of the natural resources management that was issued by Korean government that controls the genotypes or the phenotypes of the rice and soybean. This is just an example. And this kind of management has a value of millions and trillions of the money. So having the reference genome is really the start of this unlimited possibility or building enormous value chain. So how are we going to do that in scientific terms? Yeah, these are the lists that you need to get into and conquer. Like starting from identifying SNPs or QTLs or the parentages or many other things. So these are the things, you can do these things very easily if you have many genomes fully sequenced or sample. It's no problem. It takes a lot of computing power, but that can be done. All the data is there. But the problem is with just one reference sequence, you cannot do it. You have to use multiple samples. So it's like, how are you going to do that? We can do the de novo sequence for all the samples that we are interested in, but it is too expensive and it'll take forever. The second part is resequencing individual samples with a very good reference genome. And this is actually the adopted strategy for the human application. It's, for example, the clinical applications. But the price is going down, but not yet there. And I would say it'll take about 5 to 10 times more expensive to do the whole genome sequence, depending on the size of the genome and the completeness of the reference. So the researchers have been working on the bypasses so that they can get away with less sequencing. Yeah, that's totally understandable, and that is a very, very important applications. So the first thing they the Genomics Society studied was they decided to sequence only some part of the genome, not the whole. So this is not the first thing, but it actually came from the epigenomic studies, which first introduced the reduced representation library. The second one is, why don't we just pull the samples and get only the population level data? But obviously, you don't get any individual level data, which is any different applications. So there you go. We are looking at the low coverage sequencing of the whole genome in LC-WGS. This is the rest of the session. I have used a review article written on the bottom right. And it is a very, very comprehensive guideline for anybody who are interested in the genomics applications in plants and animals or even any kind of genomes. For the reduced representation, which you use is different samples, different libraries, but only uses the restriction enzyme, and you only sequence around the restriction enzyme site. So you are basically having multiple and single library data, but you are missing a lot of other part of the genome. So that is the the fundamental flaw of the reduced representation libraries. So you end up missing a lot of important markers. For the full sequencing, you can get the full coverage, genomic coverage. You can get high depth coverage, but you don't have any individual data because you pulled all the samples and you work with only one library. So it has inherent limitation. But the LCWGS is sequencing actually individual libraries for the whole genome, not just part of the genome, but very low depths. So this is the base, the theory about the low coverage whole genome sequencing. The first thing, cost. Sequence cost is not too much because we are sequencing like 1, 2, or even probably up to 8 depths X. So compared to the resequencing, which we usually do 10 to 30 to 60X. So it's much less sequencing. But the library cost is less scalable. So there are many different companies who are working on this one. I won't mention those companies, but their work is actually they have paved the ways to the low-pass genome sequencing. So the per sample cost is not very much. The second one, what is the basic requirement? You should have a reference genome. If you don't, you should have a reference genome of closely related species. Or if you have a reference transcriptome, or RNA data, that will also great helpful. And you are going to use a lot of different analysis to bioinformatics, which has been, they expand. But they are coming up with many new and more efficient pipelines these days because LCWGS is getting more and more popular. So, the basic question and this paper throws to us is how to make a perfect experimental design. So that means how much data we need to generate the sequence, the data, and how many samples we need to get enough population data, and how expensive is the experiment and the analysis. The more, the better. And it is still applied widely in practice concept. But the main question is the population genomics approach application is more sample or higher depths. And it also comes up with the how deep is enough and how many samples are enough. And the next a few slides are the tests they had done to answer these questions. So these are allele frequency estimation. On the x-axis, these are the number of samples. On the y-axis, these are the depths from 0.25 to 8. And as you can see, higher depths and more samples, you will get much better, exact allele frequency estimation with a lot more, the definable allele size. And the second one is the spatial population structure. It is, that simply means that you are working with very closely but distinctive population, and you are interested in how you can, replicate their distinction from the genome data. As you can see, with the low migration scenario, that means that this population doesn't really communicate and share their genomes. This one is that these populations are actively sharing their genome. So with the actively sharing genome, with the lower sample number and lower depth, you cannot really tell among these populations. But with the higher depth and with the the higher coverage, you will definitely get the original picture. And this is probably is the most important. So the divergent selection, that is in other words, identifying the SNP markers. And if you can take a look at the x-axis, of course, it'll get better with the sample size for population. It is more important because the marker is with lesser sample number, you won't be able to get good marker because the SNPs has a lower allele frequency. So with these numbers, only SNPs that you can get is the not really SNP. So with the higher depths, it improves, but not much. So basically, if you think about the sequencing output is the multiplication of number of samples and depth of sequence. For the most applications, the number of samples is more important. That means you sequence more samples with a lower depth. For the allele-dependent matrix and linkage disperium, high depth is preferable than the many samples. So that's actually one of the conclusions of this paper.
Dr. Young, pardon, you have around 8 minutes left. Thank you.
Thank you. So this is actually the replication of the story in the previous slide. So if you are looking for the number of SNPs, the more depth of coverage definitely helps. The more you sequence, the more SNPs you can find. But the divergence peak, you can you don't really have to go higher, like a 8x, 6x or 8x. The difference between 2x and 8x is not much. And for the population structure, this is the diagram of a principal component analysis. They don't, they don't differ much. 0.5x and 8x, you don't see too much difference. So these are the pictures of the low-pass sequencing. So you can decide how many samples you need to see to find what the exact information you want to get. Okay. So that was all for my the plant genome sequencing session. And with the time left for me, like 5 minutes, I will briefly introduce Macrogen's product portfolio. These are the sequencing platform Macrogen has. We have a world top 5 capacity for the sequencing and data analysis, including the non-profit organization. Currently, we have 15 Nova 6, many different smaller Illumina sequencers, Promethean from Oxford Nanopore, Irontron from Thermo Fisher, PacBio, RS2 SQL, and 3730XL. These are the list of products in the CES sequencing and NGS sequencing. So there you go. We also have the low-pass sequencing product. We are, we started out with the serving the research market, but we are very active in clinical sequencing service as well. And so for that, we have acquired a clinical laboratory in United States and get a CLIA and CAB accreditation in Semoga, and we got CLIA and CAB accreditation for HQ, and we also have CLIA accreditation in Singapore. And these are the selected number of the clinical applications. Which this is the plan is for the family planning. FAST is the the prenatal screening. BUBA is the postnatal screening products. That's not all. We are more dedicated to the cancer genomic solutions, clinical solutions, because cancer is basically the disease of the genome mutation. So we have a somatic cancer panel, which is actually pan-cancer panel, selected cancer panel, liquid biopsy cancer panels that we are serving many different clinical markets. And we are also having the non-patient and consumer hereditary cancer panel screening products as well. Among this one, our bestseller is the BRCA testing. So for the personal genetic service, we have a consumer side product called My Genome Story, and we have a little bit more, the high-end side with the premium genome analysis with our brand name GenTalk. It detects many standard cancer disease markers. And the other personal analysis service is microbiome. We are actively marketing the microbiome business in Korea and also in the United States. And we are working on the higher solution, the biome, which we are going to use in a medical and the health related purposes. We also synthesize the oligos, and we have many different platforms for the microarray, including Illumina, Epimetrics, and Agilence. And finally, but actually, Microgen started out with this business, genetically engineered mouse. So it's been more than 25-year-old business. We make the transgenic mice or knockout mice. We do the services. We provide the animals, and we also provide the R&D strategies for using this genetically engineered mouses. So this is the end of my presentation. So thank you for listening, and I hope you will be useful for your research. Thank you.